My name is Varish Barama, and for my fifth period project, I'll be talking about the Operon, the LAC, and the TRP. So if we go to the next slide, this is the first model of the LAC. So you can see the glucose and the lactose like trying to go in, and you can see all the new things, and I'll explain it right now. And this is the early state and the observations that I made, so I was looking at it. So you can see the LAC Operon, you can see how it's coded, you can see the mRNA, the repressor, you can see how the LAC mRNA is over here. And you can also see the PIPs, everything right here is like, you can just like, yeah. And let's start with the first info. So without lactose, the LAC Operon is blocked and can't function the way it's supposed to. Here, the repressor protein of the regulator G links itself tightly to the operator in the region of the function. As RNA polymerase travels along the DNA, this prevents it from going through transcription and letting it like transcribe the structural genes. When this like fails to occur, neither mRNA nor the enzymes needed to break down the lactose can be produced. So this prevents the cell from wasting energy on the making of enzymes when lactose is not an available food source. So let's talk about lactose and when it gets inside the cell. So the LAC operon gets started when lactose becomes accessible. By this link to the repressor protein, the lactose, more like it's like isomer molecules, becomes an inducer. So this repressor structure changes by its binding, making it unable to adhere to the operator. When repression protein doesn't work, RNA polymerase binds to this thing called a promoter, which transcribes gene structures. This can tell the cell that the mRNA can be translated into a lactose that breaks down a protein called B-galactosidase. This cell can now be can use lactose as a fuel for like energy and things like that. So now let's talk about lactose getting converted into an inducer. So when the LAC operon becomes active, it renders the repressor inactive. The allosose, the inducer, can attach itself to the protein and change the shape of the structure. This modification can help the repressor from connecting to the DNA of the operator's region. The DNA polymerase can also still transcribe the genes if the operator is still unblocked. So let's talk about transcription now. So when the LAC operon stops working, the repressor isn't really blocking the operator anymore. Because of this, RNA polymerase can still bind to the promoter part of the DNA. This is when transcription starts. So during this process, RNA polymerase walks along the DNA template, making strands and doing all the RNA stuff that basically just makes another copy of all the stuff. So you can see the CAP site, you can see how it promotes RNA polymerase binding, and you can see the repressor blocking the RNA polymerase from the operator. And this is the TRP model, so you can see the TRP mRNA, you can see the TRP repressor going into the TRP and like binding to it. And this is the first stage, and this is the explanation of that first stage. So in a system without TRP, the TRP is always going to be on the protein that can properly bind without an operator. This lets RNA polymerase begin and start to contrive the genes that can turn into enzymes that can make a TRP. Examples of this are like the TRP A, B, C, D, and E, which are showed to help with that. And this is when TRP fully starts, when it starts. So the first starts when TRP starts to become abundant. This can become a co-repressor that can bind to an inactive protein. The active repressor is now able to link with the DNA and lets the operon prepare to shut down transcription, starting a negative feedback loop and regulating the end of the link. So these are some background questions. Okay, so first of all, what is a gene? A gene is a part of a DNA strand that is transcribed into RNA and can be translated into a protein that determines the trait of a cell. What is the role of mRNA polymerase in protein synthesis? mRNA polymerase binds to the promoter. This lets the mRNA this helps it make mRNA by reading DNA during something called transcription. What is the role of ribosomes in protein synthesis? Ribosomes start the building blocks of translation and assemble amino acids into bigger proteins called polypeptide chains. What are the roles of an operator, promoter, and structural genes within an operation? Operators help block transcription, promoters start to begin transcription, and structural genes that can code for other proteins that help for metabolism. How does the presence of absence of lactose affect the lac operon? So when there is no lactose present, it starts a system that ends with the repressor binding to an operator, turning it off. When it's present, this repressor changes and releases into an operator that turns it on. And there's actually, if there's no TRP, the operator's on, and when there is TRP, it's off when the TRP is present. And what is a regulator gene? It's basically a gene that lets the portion code, and it basically chooses if an operon can be on or off. It's pretty cool. And these are some analysis and conclusion questions that we have to explain. So, let me pull up the questions. Okay, so when it gets activated, it starts to get transcribed into mRNA. So this basically makes it so that the protein production, and it's like the opposite when it's not activated. So in TRP, the repressor protein becomes active when it's connected to the co-repressor. And in LAC, allolactase can act as something called an inducer that turns the operon on. A mutation could cause an operon to stop working, doing the opposite it normally would. For example, it could keep the operon off forever, or cause the operon being oppressed and changing, leading to a lot of bad things. Without an operon, the transcription can never start, so no mRNA would be made, and the operon would be permanently off. This is what happened during it. Yeah. And when tryptophan levels are high, TRP can bind to the repressor, shutting it down. This is essential because it prevents overuse and wasting resources. These can both maintain homeostasis. Thank you so much for listening to my presentation.